talk a little bit more about protein structure. The reason this is so important is that a 3D structure of a protein is essential to that particular protein's function. We know the exact sequence of amino acids that this protein should be. After translation, it starts to fold up pretty much immediately upon leaving the ribosome. And it's going to assume these higher order structures. It may interact with other polypeptides. But again, that 3D structure, that 3D conformation of proteins, absolutely essential to that protein's function. So let's focus in a little bit first on amino acids. So amino acids are all going to have several groups in common. They're all going to have a carboxyl group, or C double bonded to an O with a COH on the other end. It's going to have a central carbon that's attached to a hydrogen. And it is going to have an amino group, which is a nitrogen bound to two hydrogens. It is also going to have what we'll call here an R group. You can call this a radical radial group, rest of the molecule. Okay? They will often use this in shorthand just to indicate rest of the amino acid. And every amino acid of the 20 amino acids is going to have a slightly different structure here. So what's going to happen as translation proceeds is you'll have a series of reactions. We're going to call them dehydration reaction. And what's going to happen is this hydroxyl group, the OH group here, is going to combine with the hydrogen from this nitrogen. It's going to combine and hopefully make a very familiar molecule, H2O. It is going to be removed. This carbon and nitrogen are then able to bond directly together. So now you have this C double bonded to an O, this carbon bonded to the nitrogen, this hydrogen still hanging around. And we're going to call this a peptide bond. The R groups, so we're going to talk about those now. They're going to give it very specific chemical properties. So you don't have to memorize the structure or anything like that for this particular class, although I'll warn you, if you do go on, you take classes like biochemistry, one of the first quizzes I ever had in biochemistry was to memorize the structure, the 3-letter code and the 1-letter code for all 20 amino acids. That was our first quiz. So it's worth looking at least looking at many of these. So they're going to be able to be grouped into those that have similar properties. Some of them are going to be nonpolar. These are typically going to be hydrophobic. They may find themselves tucked inside the center of a protein, or they may find themselves in a very hydrophobic place, like maybe the plasma membrane. Some are going to have, we're going to call these aromatic side groups, or these large ring structures attached to them. Some of them are going to be polar, but uncharged with these side groups. Polar, another word for that, would be hydrophilic. And then some of them are going to have either a positive or negative charge to their side groups. And you could imagine that these might be attracted to one another, so they might be important for the 3D conformation of a particular protein. And these polar or hydrophilic regions might be important for either on the outside of the protein if it finds itself in a cytoplasmic environment or an aqueous environment. It might find itself tucked into the center if it's in a hydrophobic environment, such as the plasma membrane. But again, it's going to allow the protein to form its 3D structure and interact with other molecules. This peptide bond, again, forming by this dehydration reaction, you're just having one amino acid attached to the second amino acid. This interaction we call a peptide bond. There's always going to be an amino end and a carboxyl end, the COOH and the NH2. So when we're talking about protein structure, there's 4 potential levels of protein structure. The primary structure is just simply the sequence of amino acids. If you were to think of them, say, as beads on a string, the primary sequence of that particular protein. As it begins to fold up, as it begins to interact with one another, these different amino acids, you start to get the higher order or higher levels of this protein structure. And the 3D structure of any protein is just a product of its primary structure. So if we're looking at some typical secondary structures, it's typically going to form either this alpha helix or a beta sheet. And what you can see here are many different hydrogen bonds, and they're just different molecules that are going to be interacting with one another. In this case, the carboxyl and the nitrogens are going to be interacting with one another to form this helix or this particular sheet. An example of the tertiary structure is just, for the most part, the entire polypeptide and how that would fold up. So in this case is an example of myoglobin, which is a respiratory pigment. You can see here many different alpha helices. Basically, this is being represented as a ribbon structure. But you could imagine these individual atoms, carbons bounded to one another here, carbons and the nitrogens for the backbone of this particular peptide. In the quaternary example, and not every protein complex will form a quaternary structure, but in this you're going to have several different polypeptides that are going to be interacting with one another. So in this case, several different genes, each coding for its own unique polypeptide, interacting with one another to form one larger complex. And the example here would be hemoglobin, and you've got central heme groups, and you've got, in this case, 4 different polypeptides interacting together. Determining the 3D structure of proteins is incredibly important, and there are many different research groups that are focused on determining the structures of proteins. The reason for this is severalfold. First of all, many proteins involved with disease arise because they're basically misfolded proteins, or proteins that are folded incorrectly. The reason they're folded incorrectly, well, usually because you get a mutation so that these interactions can't happen. The other reason is by looking at these protein structure, you can determine what the active site of a protein is. In this case, the active site of the protein looks like it's localized right here. When you're looking to design a drug to treat different enzymes, knowing what the active site structure looks like is very important for designing a drug that's going to fit very well for that active site and be a good inhibitor for that particular enzyme.